Hello guys and welcome back to another video. Today we're looking at a very interesting histology slide. The slide of a pancreas, which is a classic exocrine gland that can be easily mistaken for the parotid salivary gland except for one key feature, the pale clusters known as the Islets of Langerhans. These endocrine islets distinguish the pancreas from any salivary gland, so they are very important to recognize and understand. Now let's talk about the exocrine organization, the lobules and the ducts. Like other exocrine glands, the pancreas is divided into lobules. These appear as clusters of darker staining tissue separated by a thin connective tissue septa. Now here is an example of a large lobule, a smaller lobule, and a tiny lobule. Now within these and between these lobules we see ducts. And they come in two main categories, and it's very important we understand the naming of these. Number one, the interlobular ducts. These are simply inside the lobules and surrounded by acini. And number two, the interlobular, also known as the extralobular ducts, which are between and surrounded by connective tissue. Now, within this, you might also spot large blood vessels nearby, such as large veins, arteries, smaller vessels, and ducts with tubular or columnar epithelium. Let's talk about the islets of Langerhans. These pale staining clusters contain endocrine glands, such as alpha, beta, delta, etc., that secrete hormones directly into the bloodstream. Now, it's very important to know that you cannot distinguish between alpha from beta cells in routine H&E staining. So simply identify the structures as an islet which is lighter, less granular, and not organized into acini. Let's talk about the exocrine acini and the zymogen granules. The bulk of the pancreas consists of exocrine acini. An acinus is a small cluster of 5 to 10 serous packed with zymogen granules which stain, dart, and fill the apical cytoplasm. Features of the acinus cells are granular cytoplasm containing digestive precursors, which are enzyme precursors. Nuclei push down towards the periphery, and they are organized into small berry-like clusters, the acini. Let's talk about the Centroacinar cells. Sometimes you see these pale, what almost look like fried egg-looking cells in the middle of the acinus. These are called the Centroacinar cells, the beginning of the duct system. The characteristics of the Centroacinar include pale cytoplasm, no zymogen granules here, and they are located at the center of the acinus. And these represent the start of the intercalated ducts. Let's talk about the interlobular and interlobular ducts. Now let's identify a clear duct example here. Here is an interlobular duct, which is found between the lobules, surrounded by acini, a thin wall made of cuboidal or columnar cells. It has minimal connective tissue around it and is often called intercalated ducts when they are very small. Let's look at an interlobular duct, which is found between lobules, and this is surrounded by connective tissue, not acini, lined by more obvious epithelial wall, and they are larger in size. Let's quickly look at other structures which are commonly found in the histology side of a pancreas, such as fat cells, which are scattered throughout here. Here is an example of some nerves, which are seen as small round bundles, some arteries. You can identify these with the thick muscular walls, and the veins, which are thinner walls, and the walls are a lot more irregular. Now, in summary, on this single slide, we can confidently identify exocrine lobules now, interlobular and interlobular ducts, acini packed with zymogen granules, centroacinar cells, islets of Langerhans, which are endocrine, and arteries, veins, nerves, and fat. Once you recognize these components, the pancreas becomes one of the easiest ones to identify on the histology slide. And always remember, exocrine, 2 ducts, 2 islets. Now that was a complete overview of the slide of a pancreas, talking about histology slide here. And let's move on to the next very exciting slides on this playlist. See you then.